Have you ever wondered what makes your bread rise and your beer bubble? Meet Saccharomyces Cerevisiae, also known as baker's yeast, the microscopic mastermind behind baking and brewing production since at least the New Stone Age. This is thanks to the yeast's astonishing ability, termed the Crabtree effect, that lets it anaerobically ferment sugars even in aerobic conditions. More recently, S. cerevisiae has donned a lab coat and established itself as a powerful scientific model, helping us unravel biological mysteries such as aging, cell cycle control, and disease. It was even the first eukaryote to ever be fully sequenced. However, despite decades of research, many mysteries about the yeast remain unsolved. S. cerevisiae was assumed to be a mere product of domestication, only surviving in breweries or bakeries, until researchers made a rather shocking discovery. Domesticated yeast has had wild counterparts living in untouched primeval forests all along. Since then, scientists have wondered whether the evolutionary origins of S. cerevisiae can be traced to temperate forest habitats. Ecologists are currently exploring the yeast's natural environments, aiming to unveil the ecological factors that have shaped yeast evolution. However, finding the answer has not been easy. Wild Saccharomyces strains appear ubiquitously distributed across several forest habitats, most notably including tree bark, soil, leaf litter, and fruit. So which of these are the true ecological habitats of the yeast? Let's delve into some proposed theories. Are yeast adapted to sugar-rich fruiting environments? This theory makes sense. After all, in winemaking, S. cerevisiae thrives by fermenting sugars. Producing ethanol may also confer a competitive advantage, hindering the growth of other microorganisms. However, real-world fruit environments are very different from the controlled environments of winemaking. In these environments, fruit mixtures are confined, preventing ethanol evaporation and limiting oxygen exposure. But these conditions don't naturally exist on wild fruit surfaces. Plus, fruit is ephemeral. If it were the yeast's primary habitat, how would it survive through winter? One theory is that the yeast forms resilient spores on the forest floor during the colder months, awaiting spring where insect vectors can carry the yeast back to fruit. But this was challenged by findings showing that the yeast is astonishingly rare on natural fruit surfaces, making up less than 1% of the fungal population. If S. cerevisiae was truly specialised for fruit environments, surely we would expect to find it in much greater numbers. So perhaps yeast is not niche adapted to fruit after all. Surprisingly, yeast is most frequently isolated near oak trees. However, not all oak real estate is equally desirable. S. cerevisiae has been shown to poorly grow on oak bark extract, suggesting that oak bark is likely a poor niche candidate. However, oak soil and leaf litter seem to be hotspots for yeast assembly, with yeast decreasing in abundance with distance from the oak tree. Now, this is where things get even more intriguing. Saccharomyces is not found consistently across all oak trees. So what ecological factors are making certain oak trees more suitable habitats? And here's the big question. Are the researchers focusing on oak trees simply because they know they can isolate yeast there? An alternate hypothesis suggests that the yeast evolved for a more nomadic lifestyle. Essentially, this means that the yeast can live in several different habitats, instead of calling just one place home. This may explain why it's found at such low abundance in any of its proposed habitats, such as fruit or oak trees, as it's never really adapted to any of them. Backing this idea, the yeast can survive in several conditions, such as low and high pH, or within a wide temperature range. This versatility may be key for survival in several environments, and therefore, could this be more evidence that the yeast has evolved as a nomad? By studying the natural habitat where S. cerevisiae thrive, we can uncover the environmental conditions that support its survival, and also the stresses it has evolved to withstand. But that's just the beginning. Exploring where wild yeast assemble may also lead to the discovery of novel strains that have unique properties we can harness. Wild strains tend to be genetically diverse, often possessing unique properties that set them apart from commonly used industrial strains. Some strains could introduce new and exciting flavours to beer and bread. Others may enhance fermentation efficiency, improve stress resistance, or even revolutionize biotech applications, from biofuel production to medical therapies. Beyond S. cerevisiae's industrial potential, understanding its role in natural environments, including through interactions with other microbes or plants, or even insects, could reveal symbiotic or antagonistic dynamics. These findings may have broad application in fields such as forest management and agriculture. And so, although we don't have all the answers yet, the possibilities are so vast, and each discovery may bring exciting opportunities. And so, the search continues.